I'd like to start by just thanking the organisers for inviting me to talk today. I've really enjoyed the session so far, and I'm very much looking forward to the talks to come as well. And I'm really pleased to be able to speak about the last nearly 15 years of research that we've been conducting as part of the Gemini twin cohort. So I thought I'd start by explaining why it's useful to understand the role of genetics in behaviour, because sometimes in behavioural genetics research we get criticism that essentially it's blue sky thinking and that there's very little application of some of our work, and potentially even identifying genetic contributions to health behaviours or obesity related behaviours simply can promote the feeling of fatalism, or essentially the idea that someone can't change their behaviour. But so there are multiple reasons why it's useful, or I believe that this kind of research is extremely useful. So one important aspect is there's a lot of blame and guilt around obesity and health behaviour, especially for parents when we're considering these behaviours in children. And identifying genetic contributions to some of these behaviours helps alleviate that blame to some extent, and identifies reasons beyond potentially the control of the individual. It also can help identify windows of opportunity for intervention. This kind of research can inform where best to target environmental intervention, so I'll explain a little bit more about that as I go through my talk. And it's also the first step towards molecular genetic research, identifying specific genes involved, which we've heard a little bit about earlier today. So human body weight has a strong genetic basis. We've already heard about this today, but I just thought I'd share this image because I think it quite clearly illustrates in one slide the genetic contribution to body weight. So identical twins on the left and non-identical twin pairs on the right, and you can see the striking similarity in body weight between the identical twins compared to the non-identical twins. And this is of course a strong indication that there's a highly heritable component to body weight. And in fact, meta-analyses have shown that there's the heritability of body weight is somewhere between 50 to 90% in both children and adults. But if we know that body weight is highly heritable, we need to understand a little bit more about the mechanisms involved in the development of weight as a result of our genetic inheritance. So there's three potential pathways through which genes could be influencing body weight. That's through the partitioning of nutrients into fat, through energy expenditure, and through energy intake. And in our team, over a decade, we've been developing an appetite model of obesity. So this is called behavioural susceptibility theory. And the idea is that genes are influencing energy balance and weight gain via, at least partly, via the neurobiology that controls appetite. So that individuals are inheriting a propensity to overeat in response to their environment, and this in turn leads to a positive energy imbalance and weight gain. And at the heart of behavioural susceptibility theory is the idea that genes interact with the environment to lead to weight gain, so that people are only able to express their inherited appetitive phenotype if the environment allows them to do so. So when I'm talking about appetite in the context of our research, we tend to focus on two main aspects of appetite. So food responsiveness, which could be considered the on switch for appetite. So the idea that when you see or smell really palatable food, or you see someone else eating highly palatable food, the strength with which you desire to eat. So some people will walk into Pret-a-Manger and buy a coffee and be able to ignore all the delicious cakes on display, while others will be highly cued to buy them and eat them because they maybe are more food responsive, more have a more avid appetite. Satiety responsiveness is the flip side, so that's more like the off switch of appetite. So that's the idea that some people find it easier than others to stop eating at the point in their fall, or they're more able to recognize their own internal feelings of fullness. When we're looking at these appetite traits in twins, this enables us to tease apart the contribution of both the genes, both an individual's genetic inheritance, their genes, and the environment to the individual variation in these traits. So I know that there's going to be more discussion of twins later today, but I just thought I'd show this for anybody that's not highly familiar with the twin method. So not only can we quantify the contribution of genetics to individual variation in a phenotype, we can also tease apart the shared environment and the non-shared environment contributions to that trait. And that can be helpful because that can indicate where interventions might be better directed. So in our team, we study appetite mainly in paediatric populations, and there's a number of advantages to this. Children are less susceptible to social desirability. So in early life, they don't understand what's considered healthy, and they're not going to adapt their responses or the behaviour to researchers. They're unlikely to be dieting in the early years. They have, just by virtue of their age, less long-standing obesity. And also, if we can examine these relationships from the start of life, it is easier to tease apart cause and effect in the relationships between obesogenic behaviours and weight development. So Annika mentioned that I'm the deputy director of the Gemini twin cohort. So Gemini was established in 2007 by the late Professor Jane Wardle, and it was established to look at the relationships between appetite and growth and other obesogenic health behaviours from the very start of life. So the twins are turning 14 now, but we've collected a multitude of different data from these families at various points over the past 14 years. So I've just put this slide up really. I'm not going to talk to it for very long, just to show that some of the variables and data that we have in these families. And I'll be speaking about some of that today. So alongside various questionnaire measures, dietary intake data, DNA, we also collect anthropometrics, height and weight from the families at 3 monthly intervals since birth. So I thought I'd talk through some of the research that kind of supports behavioural susceptibility theory that's been conducted within our group, our team. And the first step is really to establish a relationship between appetite and weight in childhood. So Jane, along with Susan Carnell, developed the Child Eating Behaviour Questionnaire in 2007. So the CEBQ, or Child Eating Behaviour Questionnaire, is a parent report questionnaire that measures variation in a number of different eating behaviours or appetite traits in childhood. And again, the ones I'm going to be talking about are food responsiveness and satiety responsiveness. And you can just see some examples of the types of questions parents respond to, and on a Likert scale. So this allows us to collect quite a lot of appetite data relatively quickly. And it's actually been shown to be quite reliable. So there has been kind of behavioural validation studies of the appetite traits measured through the CBQ, and they've shown it to be both valid and reliable. But also more recently, in the last year or so, we conducted a large meta-analysis of appetite and BMI in children using the CBQ, which has now been cited over 400 times. And we identified 72 studies that looked at relationships between the CBQ and weight. And what we found was that food responsiveness was reliably associated with higher BMI in children, and increased satiety responsiveness was reliably associated with lower BMI. So children who were more food responsive were heavier, and children who were more satiety responsive were lighter, which is what we would expect, but it's encouraging that the data supports this. However, the issue with identifying cross-sectional relationships between appetite and weight is that we know that there's likely bidirectional associations going on, and as an individual's body weight increases, their appetite may increase as a result of their increasing body weight. This would make sense. So it was also important to establish prospective or causal relationships between appetite and weight. And we were actually able to do this in the Gemini cohort, because when the twins were 15 months old, we used data from their weights from birth, essentially. So we used a baby eating behaviour questionnaire. So this is the infant version of the child eating behaviour questionnaire. and we were able to identify discordant non-identical twin pairs for the two appetite traits that I'm speaking to today. So for satiety responsiveness, we found discordant pairs, so that were 1 standard deviation different in terms of their satiety responsiveness score. And we looked at their weight trajectories over the first 15 months of life. And what you can see here is that the more satiety responsive twin, which demonstrated by the lower line, at 15 months old, weighed 1 kilogram less than their less satiety responsive co-twin. And that might not sound much, but actually at 15 months old, that's 10% of your body weight, or 10% of their body weight. So that's actually quite a substantial amount. And the same was shown for food responsiveness. So discordant twin pairs, the more food responsive twin weighed 1 kilogram more than their less food responsive co-twin by 15 months old. And while we did identify the reverse relationship, the relationship from appetite to weight was much stronger than the association from weight to appetite. So this does suggest that appetite is playing a causal role in weight gain in the early years. Now, interestingly, since this study, there's been several other studies in older children looking at prospective relationships between appetite and weight. And again, bidirectional relationships are observed, but there's some indication that the relationship is stronger from weight to appetite in the later years. So there's much more research needed in this area to fully understand these quite complex relationships. So next step in terms of testing behavioural susceptibility theory is to establish that appetite has a genetic basis. And again, using the baby eating behaviour questionnaire, the infant version of the child eating behaviour questionnaire, the brilliant Dr. Claire Llewellyn managed to establish that food responsiveness and satiety responsiveness are both highly heritable in early infancy. So you can see the genetic effects in the dark blue bars here. Food responsiveness is on the left and satiety responsiveness is on the right. And both for both appetite traits, the percentage of variance explained by genetic effects is high. There is also a contribution of shared and non-shared environments as well. But what's quite surprising about this strong genetic component at this age, I think especially, is the fact that people have long believed that children are born with an innate ability to control their own appetite, essentially. So that if parents feed infants responsively in accordance to their feeding cues or their appetite cues, that all children are born with the ability to stop eating when they're full, essentially, to display very good satiety responsiveness. And what we're finding here is there is quite a wide individual variation, and that this variation is largely explained by genetic differences. So even from the start of life, appetite is showing a strong heritable component. So we've done similar studies for other obesogenic behaviours, and I just thought I'd speak very briefly about food preferences because this is another interesting one. So we used, again, a questionnaire to measure food preferences in the Gemini cohort when the twins were 3.5 years old. So parents reported the extent to which their twin children liked a number of different foods, and then these were grouped together through a principal components analysis into essentially what we would consider established food groups. And we were able to estimate the quantify the contribution of genetic and environmental influences to variation in liking for these different food groups at 3 years old in the Gemini twin cohort. And what we found was that the genetic contribution to the more nutrient-dense foods, so fruits, vegetables, and proteins, were higher on the whole than those for the more energy-dense foods, so dairy, starches, and snacks at 3 years old. And what we also saw was quite a strong influence of the shared environment across all these food groups, but particularly the energy-dense foods that we might want to curb liking for. So in a way, this is encouraging because it's suggesting that you can manipulate the environment to influence the extent to which children are showing preferences for these foods in early life. And Andreas Smith repeated the study in the TEDS twin cohort when they were in their late teens, when they were around 19 years old. So obviously this is a different cohort, so direct comparisons need to be cautiously considered. But essentially, the pattern of genetic influences on liking for the different food groups was very similar at 19 to what we found in the Gemini cohort at 3. But what we did see in the TEDS cohort was that the influence of the shared environment seemed to have disappeared completely by this age. So again, that's not surprising, because by this age, the twins may not even be living together. Their influences will be broader, and they were spending more time outside the home. But it's also interesting from when considering interventions, because it's unlikely that interventions to promote the liking of nutrient-dense and maybe dampen the liking for energy-dense foods would work if they were targeting the home environment at this age. So fundamental to the behavioural susceptibility theory is the influence of the environment, and obviously that's what this session is also largely about. So we don't exist in a vacuum. Our genes have to interact with the environment in terms of how they're expressed. And there's obviously going to be environmental influences on obesogenic behaviours and appetite. So a good example of a way to characterise the obesogenic environment is through measurements of deprivation, because we know there's quite a strong socioeconomic gradient in terms of obesity prevalence, and this can be seen quite clearly in children. So this is data from the National Child Measurement Programme in 2018 to 2019. So for those people that don't know, all children entering and leaving primary school are weighed and measured, and their BMI is calculated. And this is data showing that obesity rates in the most deprived decile of children, both in the first and the final year of primary school, are almost double that to the least deprived. So this is really stark. We know that the genetic makeup of children in the most deprived decile will not vary substantially from those in the least deprived. So these differences can only be explained by differences in the obesogenic environment. We wanted to look at relationship between the obesogenic environment and appetite in Gemini. And again, we used SES as a proxy. And we used a composite measure of SES in the Gemini families that took into account a number of different variables that are known to tap into socioeconomic status. And this work was conducted by Alison Fildes as part of her PhD. And what she found was that at 16 months, lower socioeconomic status was associated with higher foods responsiveness and lower satiety responsiveness in twins from the Gemini cohort. So what we were seeing were that children children from more deprived backgrounds were more likely to be food responsive and less likely to be satiety responsive, or have lower satiety responsiveness, at this age group. And repeating this at 5 years, the relationship was still there for food responsiveness, although it disappeared for satiety responsiveness. So looking at this longitudinally or prospectively, Alice demonstrated that lower socioeconomic status predicted greater increases in food responsiveness from 16 months to 5 years. So the children growing up in lower socioeconomic status households showed bigger increases in their food responsiveness, so the extent to which they displayed avid appetite traits compared to twins growing up in higher SES households. So this demonstrates the influence of the environment, essentially, on the development of appetite and weight. But we wanted to look at specifically gene-environment interplay in the development of obesogenic behaviours and body weight. And we were able to do this in Gemini because of the genetically sensitive design, and because we have collected quite a large amount of data on the obesogenic home environment as well, which I'll speak about in a second. So at the heart of the behavioural susceptibility theory is the idea that genetically inherited appetite traits are only able to be displayed if the environment allows it. So essentially, if you inherit a higher propensity to have a more avid appetite, to overeat, to be less satiety responsive, you can only live out these traits if you are in a permissive environment that allows it. So the more obesogenic the environment, the more that you are able to express your obesity genes, your high risk appetite genes, and that will lead to an increase in energy intake and potentially in weight gain. So in the Gemini twin cohort, we measured home environment via home environment interviews when the twins were 4 years old. This was led by Stephanie Schrempf, and more recently when the twins were 12 years old, led again by Alice Kinnaman. And this was quite a massive undertaking. So all the families were called at home, and they were asked about the home food environment, the home physical activity environment, and the home media environment. And within those three domains, they were asked about both the physical and the social aspects of those different domains within the household. So the availability of food and drinks, physical activity equipment, and media equipment, the accessibility to those different things, and alongside parental rules and the structures that controlled the use of media equipment, physical activity equipment, and access to food, and also parental modelling of some of these behaviours. So this data was used to create composite scores of the overall obesogenic home environment and also the obesogenic quality of the three domains of the home environment, so the home food, the home media, and the home physical activity environments. And as expected, higher risk home environments were associated with less healthful behaviours, so with decreased fruit and veg consumption, increased energy dense snack consumption, lower physical activity, more screen time. And at 5 years, we found that there was an association between the high risk home environment and higher BMI in the children, although this relationship wasn't apparent at 4 years old. Now, the next step within the cohort when considering the home environment was to explore the extent to which body weight or BMI was heritable according to the risk in the home environment. So essentially what we did was split the cohort into those living in higher risk and lower risk home environments on a median split. And then we conducted heritability analysis in those two groups. And what we found was the heritability of body weight in the high risk home environments was 86%. So the amount of variation in BMI explained in higher risk home environments by genetics was around 86%. And there was no influence of the shared environment in that group. Whereas in the lower risk home environment, only just under 40% of the variation in BMI was explained by genetic influences, and over a third was explained by shared environmental influences. So this supports behavioural susceptibility theory and the idea that genes are influencing weight, potentially via an appetite pathway, but they're only able to be expressed in a permissive environment. So children who inherent propensity to overeat can only express that propensity if the environment allows. So in this context, what you see is that expression leading to higher estimates of genetic influences in those children living in a more obesogenic home environment, whereas the volume control on genetic influences seems to be turned down in lower risk home environments. And what you're seeing is the influence of that environment that maybe protects against the expression of some of those inherited phenotypes, appetite phenotypes, for example. So this is really encouraging because it suggests that in early life, if you can encourage healthier environments, that you can help control someone's inherited susceptibility to obesity, and that you can reduce the variation in body weight essentially, and you can reduce obesity risk. Now, this isn't to say that parents are responsible for the development of overweight or obesity in their children. This is a very small example. Obviously, environments go much further beyond the home, and as children get older, as we saw from food preferences, it's likely that the home environment becomes less and less important in establishing or controlling some of these obesogenic behaviours. And in fact, even the concept of promoting a healthier home environment is quite problematic when we know that children likely shape their own home environment to an extent as well. So we know that a lot of the way parents feed their children is in response to their children. So we've shown in other studies that parents feed according to how their child behaves. So the children, in a sense, are likely establishing their own home environment to a degree. So I didn't want to leave with the idea that parents are responsible for the development of overweight in their children, and I wanted to try and point out that it'd be much more difficult for some children than others to control their appetite and to control their body weight, and this continues obviously into adulthood. And I thought I'd leave you with a quote from the brilliant Professor Jane Wardle, where she points out that it's actually really important to remember that blaming parents a lot is wrong, and excessive weight gain in a child is unlikely to be the fault of the parents, and is more likely to be due to the child's genetic susceptibility to obesogenic features of the modern environment. And that that suggests that we need to be thinking about targeting interventions both at the home and the wider environment, but also consider individual variation in how people might respond to those interventions. I also wanted to just point to some of the other work that's going on in the Gemini twin cohort, and if you'd like to read more about the work that's going on, please do visit the study website. We're currently collecting more data on the home environment in the Gemini families throughout the COVID-19 pandemic, to see how the pandemic has influenced the obesogenic quality of the home environment and other factors within the home and the twins. Obviously, the work I've described today has been work of a much broader group, and I just wanted to say thank you to everybody that's contributed and for them allowing me to present their work here today. And finally, to say thank you to the Gemini families for the wealth of data that they've provided over the years that's enabled this research. Thank you very much.